This protocol describes the assembly and operation of an acoustofluidic system that can efficiently deliver biomolecules for a variety of research and cell-based therapeutic applications. The main advantage of this system is that it allows rapid delivery of biomolecules to cells while maintaining their viability. It is a platform technology that allows cells to be transformed for a variety of cellular therapeutic applications, such as cancer treatment. Begin by combining 54 grams of PDMS base and 6 grams of curing agent in a cup. Mix vigorously and thoroughly with a spatula for at least 1 minute, then place the cup with the PDMS solution into a desiccator for approximately 30 minutes or until remnant air bubbles are removed. Place a photoresist-coated wafer with the patterns facing upward in a 150 mm Petri dish, then pour the PDMS solution over the mold. If needed, place the Petri dish inside a desiccator and apply vacuum until remnant air bubbles disappear. Transfer the Petri dish into a lab oven and bake for 2 hours at 60 degrees Celsius to cure the PDMS. After curing, carefully remove the PDMS from the Petri dish by cutting around the edges of the wafer with a razor blade. Cut out each individual device using a knife or razor blade, then punch holes through the inlet and outlet ports using a 2.5 mm biopsy punch. After treating the PDMS device with oxygen plasma, immediately place each device onto a clean soda-lime glass microscope slide with channels facing the glass surface. Allow the devices to bond over